Hello, my name is Demitra, and I'm excited to share my research exploring metformin as a potential therapeutic for multiple sclerosis, also known as MS. I'll begin by providing a brief overview of MS, the rationale behind using metformin, as well as our key findings. MS is an autoimmune disorder of the central nervous system that is estimated to impact 2.8 million people worldwide. It's characterized by lesion formation in the brain and spinal cord, widespread inflammation, and reductions in brain blood flow. Metformin, a commonly prescribed type 2 diabetes medication, is a promising therapeutic for MS due to its anti-inflammatory and vascular protective properties. This drug has shown beneficial effects in other neurodegenerative diseases, such as Alzheimer's disease, and was recently tested as a treatment against inflammation for COVID-19. Using an animal model for MS, we sought to determine whether metformin could slow disease progression and decrease disability, restore cerebral blood flow, and whether these changes were associated with cellular level changes. In particular, we wanted to look at microglia, which are immune cells involved in inflammation. To test this, we used an animal model called the Experimental Autoimmune Encephalomyelitis, or EAE model. This model mimics key features of MS, including an autoimmune response, lesion formation, and progressive motor disability. How we use this model is that we track disability in mice using a 15-point graded scale, which grades motor impairment based on limb paralysis. Our experiment was structured in the following way. First, we collected baseline measurements such as general behavioral testing with open field, and we also used MRI imaging with a specific sequence to assess cerebral blood flow. On day 0, we induced EAE in our mice to create our MS model. From days 1 to 28, once mice began to show signs of disability, we started treatment with either metformin or saline as a control through twice-daily intraperitoneal injections. During this period, we also monitored the mice daily using that 15-point scale mentioned on the last slide. At our experimental endpoint, all baseline measures were repeated. Brain and spinal cord tissue was then extracted and prepared for histology to stain for microglia. First, we observed that both groups demonstrated consistent and progressive limb paralysis. However, when we examined the individual disease progression plots, we found interesting results. In this figure, each dot represents a clinical disease score, and each connected line represents the disease progression of an individual mouse. We found that for all our saline controls, mice develop signs of disability that rapidly increase in severity over time with no improvement. When we look at the disease progression in metformin-treated mice, we see that the majority follow the same progression as the saline mice we just saw, but remarkably, 4 mice, which are the ones highlighted in purple, red, and orange, show an increase in clinical score, followed by a gradual decrease or plateau at a mild symptom score. It's important to note that these changes in disease progression happened after receiving metformin treatment. And since these 4 mice responded differently than the other 12 in their group, we analyzed them separately going forward, and we saw that their disease improvement was linked to changes in cerebral blood flow. In the cortex, for example, we found that while both saline-treated mice and metformin non-respondents showed a marked decrease in cerebral blood flow after becoming sick with EAE, the respondent mice, which were the ones that showed improvement in disability score, maintained cerebral blood flow levels close to their baseline measures. This suggests that metformin may have the potential to restore brain blood flow in some cases. We observed similar results in other brain regions, such as the thalamus and hippocampus, reinforcing the idea that metformin could have a vascular protective effect in MS. Our preliminary histology results also showcase a difference between treated and untreated mice. We saw an increased proportion of microglia in the hippocampus and cortex brain regions. Although microglia are typically associated with inflammation, there are other phenotypes of microglia that are anti-inflammatory. When we zoom in on the tissue, we see that there might be differences in microglia morphology between the 2 groups. These differences in shape may be indicative of differences in phenotype. While we hypothesize that metformin may potentially be shifting microglia from an inflammatory to an anti-inflammatory subtype, we would need to further investigate this with other molecular methods. In conclusion, while our study found that only a subset of mice responded to metformin treatment, the results are promising. Going forward, future studies will include optimizing the time of treatment, as our lab is currently investigating whether treating mice prior to disease onset will improve outcomes. We will also need to further investigate the phenotype of microglia present in metformin-treated mice to better understand the mechanistic underpinnings of metformin and how it exerts its beneficial effects. This could open the door to developing more targeted and effective therapies, not only for MS, but potentially for other neurodegenerative diseases as well. I would like to thank my wonderful supervisor, Dr. Jeff Dunn, for his support in this project, as well as Dunn Lab members, particularly Ying and Abby, for their help, and also the We Young Lab for inducing our EAE mice. Thank you for watching.